Welcome everybody to the GlycoNet Webinar, and it is my distinct pleasure today to introduce Dr. Parastoo Azadi, who is the Executive Director of Analytical Services for the Complex Carbohydrate Research Center in Georgia. Dr. Azadi has done amazing work over the years. She has been instrumental, I think, in really making glycomics analysis available to a much wider swath of the public and much wider swath of the biomedical research that is out there. And so she has been teaching for years. She received her bachelor's degree in polysaccharide isolation in 1987 from the University of North London, and her PhD in biochemistry in 1991 from Imperial College of Science and Technology in London, where she studied structural characterization of carbohydrates and glycomics by mass spectrometry. She was also a senior scientist at SGS, which is a mass spectrometry consultancy company, prior to her amazing work as a member of the Complex Carbohydrate Research Center. And so now I'm going to cede the floor to Dr. Azadi.
Good afternoon, everybody. Good afternoon from Athens, Georgia. I want to discuss this afternoon, give you some introduction to what we've done in the last 3-4 months on trying to understand glycobiology of SARS-CoV2, glycoproteins, and its implications for therapeutic and possible vaccine development. So we started the journey initially working on the spike protein. So this diagram shows, represents the spike protein here in the middle with this spike, the virus with the spike protein on its surface, and it's the human receptor that interacts with the virus SARS-CoV2. It's the ACE2 receptor and all the other glycoproteins that are embedded in the virus, particularly this N-glycoprotein, the nucleocaspase protein. It's another protein that I will not go into today, but is also of interest because of vaccine development. So today I will mainly focus on the spike protein on the surface of the virus and the receptor. So this is a published CryoEM structure of spike RBD and the ACE2 complex. Here in the CryoEM structure, the glycosylation sites, I've just added the glycosylation sites here for the ACE2. The glycans that was published by the CryoEM, these are just arbitrary glycans. They had just put some maybe disaccharides or monosaccharides there in the place of the glycans because there was no actual structure determined for the glycans. But as you will see this afternoon, we've gone ahead and done characterization of these individual sites and the glycans on the receptor. So what this shows is actually, this is the receptor binding region of the green is the receptor binding regions of the SARS-CoV2 that it interacts with the ACE2, human ACE2 receptor in its peptidase domain region. Of interest is these regions, that glycosylation regions N90 and N322. So I'd like you to pay attention to this, and I'll talk about that later on. Another important point that I want to make a point of is the glycosylation at 0730 that I'll go into later. So these are the glycosylation sites that may prove to be important later on for these interactions. So looking at the glycosylations of the spike protein, we have decided to look at the S1 and the S2 subunit separately. We think that maybe the vaccine development will likely focus on individual subunits due to difficulties in manufacturing and purification of the full-length spike protein. And the S1 subunit is particularly because it contains the binding site, it may be of interest to the vaccine development. So, and in just looking at the individual subunits, we can avoid potential enzymatic cleavage of the protein. And viral antigens generated from various sources can influence the efficacy of potential vaccine developments. The downside of looking at the monomers versus the trimer is that there may be greater potential for misfolding of the protein in the individual subunits. But that's just that's what we decided to do, to do the individual subunits. So that's the RBD region there and the individual subunits. So what is the glycosylation of the individual subunits? So what we've done is we've looked at the glycosylation of the S2 subunit, and as you can see, we found that 3 of the sites are not glycosylated, whereas the other sites are predominantly complex type glycans. The S1 subunit, in the S1 subunit, interestingly, we have found predominantly high mannose type glycans. Specifically in the RBD regions, the N331 and the N343, we have found predominantly high mannose structures. This is in contrast to the glycosylation study that has been done on the full trimer and Max Crispin's data showing that these RBD regions are predominantly complex types. So it shows that looking at expressing these individually can have effect on glycosylation, and that could be important for vaccine development, because if they're now high mannose, if they're individual subunits, but in the actual glycoprotein, they're complex, then these are implications for the vaccine development that I'm hoping the manufacturers will take into account. The other point is that there are some 2 sites that are not glycosylated. We've shown that they're not glycosylated in S1, the N17 and the N603. And there is one site, N657, that is partially occupied. So the significance of O-glycosylation in the S1 subunits. So we have observed a glycosylation, O-glycosylation at threonine 323 in the S1 subunit. This is close to the RBD region and it's in the flexible region, so it may have implications. The structures that we have observed are 76% core 1 type O glycans and 24% type 2 O glycans, and they are predominantly silylated O glycans. So this is just a spectrum to show that we have unambiguously determined the site of Olin glycosylation on the S1 subunit using the fragmentations, MSMS spectra, and diagnostic fragments for both the total mass and the fragment mass for the particular glycopeptide we were looking for. So what about the overall glycosylations of the different subunits? So in the S2 subunit, the 1098 site is very heavily glycosylated with larger N-link glycans. Overall, I would say the S2 site is more complex, as I said, but also heavily glycosylated on those sites that are glycosylated. The S1 site is predominantly high mannose. There is not that much silylation actually in the S1 subunit. There is more silylation in the S2 subunits. The RBD regions are predominantly high mannose. So what are the potential significance of glycosylation as a whole, but Olin glycosylation on this S1 subunit? So as you can see, this is a very flexible region, and having a glycan, O-glycan there, this hydrophobic moiety there, it's interesting to find out what is the significance of this, having that O-glycosylation there. closer to the RBD region. So another interesting observation is that this region is mutated from SARS-CoV-1. So actually, this region in SARS-CoV-1 is PSGD, whereas it is PTES in SARS-CoV-2. And they've shown that there is increased binding of SARS-CoV-2 S protein to its ACE2 relative to CoV-1. So there is increased binding for CoV-2. And it is interesting to speculate if having the O-glycosylation there close to this RBD region in the flexible region, does it have an effect on this binding increase in CoV-2 or not? So what about the ACE2 receptor? What about this angiotensin converting enzyme? And what is this angiotensin converting enzyme? Basically, it's a protein that is expressed in endothelial cells. It's important in blood pressure regulations. Basically, it splits the angiotensin to peptides, and those are important for blood pressure regulations. But how does it affect the binding and the infection? So there's one thing looking at the virus and the way it binds to the receptor, but it's important to find out about the susceptibility of the SARS-CoV-2 infections in different individuals. And also we decided to look at to see whether we can find any clues in susceptibility of infection in different animals. So there's been a couple of papers showing different susceptibility of animals to the SARS-CoV-2. But the conclusions of those papers are just the protein sequence on its own, you cannot tell whether an animal is susceptible or not based on just the protein sequence. So here in this slide, what I'm trying to present is the human ACE2 sequence up there with the glycosylation sites indicated from our glycosylation studies and pointing out the O glycosylations there also. The blue lines indicate that the animals that are susceptible, and the red line indicates the animals that are not susceptible. So if you can see, pig, mouse, and chicken are not susceptible. And interestingly, the only difference that we can see between these is the glycosylation at N90. So if you can see, if there is N glycosylation at N90, they are susceptible. But if that glycosylation is removed, then the animal is not susceptible. The N322 is not as obvious having that glycosylation there, because obviously some are there, some are not there. And actually having the actual sites of glycosylation, whether it's 7 sites of glycosylation or 6 sites or 9 sites, those are again not very clear whether those are important for susceptibility or not. The only thing that we could find that correlates well with susceptibility is this N90, glycosylation at N90. So what is the glycosylation of this a human ACE2 receptor? I like to point out that these were purified ACE2 receptor grown in HEK293 cells. So these are human cells. And what we have found, this is the glycomic study of releasing the N-link glycans, purifying them away from the protein. And what we've done here, we've permethylate them. We permethylate them to increase sensitivity, and also it allows us to do subsequent MS-MS analysis to find out about the different structures. And through the MS-MS analysis, we have found out that we have predominantly biantennary structures, some triantennary and tetraantennary, and the presence of high mannose and hybrid structures. The main components are actually this smaller biantennary structure that is not silylated or doesn't have any galactose residues on it. Half of the structures don't have sialic acid. Some have 1 sialic acid, 2 or 3 sialic acids. We've observed most of the glycans are fucosylated, and some have 2 fucose, observing both antenna and both sides of the molecule have fucosylations. So this is the glycomic study. Just the glycomic study is not going to tell us which glycans are attached to which sites. So in order to find out which glycans are more prevalent at which sites, we perform glycopeptide analysis. So this is the representations of the glycopeptide analysis indicating all sites of glycosylation. The picture here on the left-hand side shows all the N-link glycosylation sites and the O-glycosylation sites. What I'd like to point out is, again, this region, N322, N90, and really N546, that is binding to the RBD regions. So the glycosylation at N90 actually proved to be, it is mostly small biantennary structures, whereas the N322 is heavily glycosylated with larger N-link structures. So it's interesting. One site that is important has a smaller glycans, and the other site has very larger glycans. The sites N53, N90, N103, N322, and N546 showed no unglycosylation, so they were fully glycosylated. Whereas the sites N432 and N690 were 26% and 1% unoccupied. I want to point out about the O-link glycosylations. We have observed 3 O-link glycosylation sites under human ACE2. The O155, O496, and the O730. I pretty much will not talk about the O15 and the O496 because the percentage of occupancy in these were less than 1%, so we don't think these are significant. This is probably just, it's an hex NAC, so it's probably just a GalNAc residue attached. But what I want to point out is the O730 glycosylation, which was fully occupied. And it is in the, what is called the cytoplasmic tail of the molecule. And so the way the ACE2 and the spike bind together is the ACE2 binds in a, it forms a dimer, and it binds to 2 spike trimeric forms. So basically the ACE2 is forming a dimer. And it is interesting to think about whether these glycosylations have an effect on this dimerization at all or not. Another interesting point is people were looking at to see whether they can use the ACE2 inhibitors to stop infection. Actually, we've kind of looked into that, and the ACE2 inhibitor is actually attaching here near the O155 glycosylation, and it's not close to the binding region with the RBD region. So it may not have an effect using the ACE2 inhibitor. So this is data showing how we've come about sequencing the individual glycans. So what we've done is we've used the MSMS, figuring out the bisecting GlucNAc present at low abundance, of course. So we've done fragmentations of diagnostic fragments to figure out the bisecting GlucNAc and differentiating between a triantennary structure, and a bisecting group NAC. So these are fragments, diagnostic fragments that shows that we can tell that there is a bisecting group NAC there instead of a triantennary structure. Again, an important point is to figure out the position of the fucose group. So looking at fragmentations, we've done, figured out the difference between core and antenna fucose. Again, looking at fragments, this 660 fragment is diagnostic of having a ion which has antenna fucose versus the core fucose. I want to point out the quantitation of these is rather difficult based on MS2 spectra. But what we can tell when we look at the spectra is if a particular N-link glycan is predominantly core fucosylated or predominantly has antenna fucosylation. But absolute quantitation of these is rather difficult just based on MSMS. It just gives you an idea that what is the relative ratio of these to each other. The other thing that I want to point out is sialic acid linkage. So other coronaviruses have shown some preference for 2,3 link sialic acid linkage. So the sialic acid also in other viruses are shown to be important. That linkage has shown to be important, whether it's 2,3 versus 2,6 has been shown to be important in other viruses too. So in this case, in the human ACE2, we've shown that the sialic acid linkage is mostly 2,3 linked. Although most N-link glycans had both 2,3 and 2,6 linked, but the N-link glycans were predominantly 2,3 linked. And it remains to be seen whether this preference, this predominant linkage of 2,3 linked sialic acid has any significance for the infection or susceptibility once further experiments are performed. So the way we determine the linkage is again by doing MSMS. So we take the particular structure, and what we are interested in is the fragmentation of that galactose residue. So we start with a biantennary structure, for example, and we perform an MS2 and obtain this trisaccharide, and then an MS3 to obtain the disaccharide, and an MS4 to obtain the monosaccharide, and the MS5 to get the diagnostic fragments. And having run standards, we know that the 2,3 link sialic acids will have particular fragments at 109 and 137, and just the relative ratios which show that the predominant fragment is coming from a 2,3 link sialic acid. So some implications of these studies, studying the glycobiology of SARS-CoV2 and these two glycoproteins. So what is our current understanding? ACE2 is the most prevalent receptor of SARS-CoV2. There are differences in susceptibility of SARS-CoV2 within species and across species. There are differences on the amino acid sequence and glycosylation sites across the species. There are differences in the amino acid sequence within human due to mutations, which is leading to altered viral binding. So I want to particularly cite this site N90 mutation. So because site N90 mutations offers a blueprint for engineering high affinity proteins and peptides that could block receptor binding sites on the S. So children express more ACE2 than adults, but adults are more susceptible. ACE2 has been seen to dramatically decrease with aging in rat models. Another interesting observation has been that the shedding of ACE2. What is the shedding of ACE2? Basically what happens is the ACE2 is cleaved and produces a shorter version of the ACE2. And this shorter version has been implicated in basically contributing to the infection. So what that means is decreasing ACE2 shedding through inhibition of ADAM17 may have a protective effect on SARS-CoV2 infection by reducing the viral load. So interestingly, the O-glycosylation site at 3,730 that we've observed that is predominantly silylated, it's at this juxtamembrane region. This juxtamembrane region is a region in the membrane that is believed to play a role in this shedding. So mutations at this cytoplasmic tail of the ACE2 could inhibit its shading and reduce viral infections. So the presence of this hydrophilic glycosylation, this bulky glycosylation at T730 in this juxtamembrane, just outside the cell membrane, could play a role in dimerizations and presenting of ACE2 on the cell surface. So we could speculate that having these bulky things up there in the in the region that is important for shedding could contribute to dimerizations and could be important for infections. There's studies being done that this juxtamembrane region, amino acids between 7160 and 741, has been shown to be important for the shedding. So could be mutations within that region. It would be interesting to do mutations within those regions and to prove to see whether once you mutate those regions, what's the effect on basically on the infections, and what is the effect on presenting ACE2 on the cell surface. So previous data has suggested that this cytoplasmic tail of ACE2 could be a candidate for targeting for anti-SARS-CoV2 therapeutics. So it will be important, I think, to take a note of these glycosylations, O-glycosylation at T730, and see where that will lead us into understanding better glycobiology of ACE2 and the way it is presented on the cell surface for the virus. So what are the questions remaining? What are some species more susceptible than others? Why are there differences in susceptibility for different individuals with species, for example, humans? Why do humans show age, sex, disease-dependent variant in susceptibility? The viral infections are usually more common in children under 5 years of age compared to adults, but why are they less susceptible to SARS-CoV-2? An individual will have the same ACE2 amino acid sequence over their lifetime unless mutations happens, but why are children less susceptible? So glycosylation study may answer some of these questions, as different species, individuals, and age groups show differences in glycosylation and glycan epitopes. Thus, it is pressing need to study glycosylation of SARS-CoV-2. And the other thing I want to point out is there is not that much known about the glycosylations of different animals, ACE2 in different animals. If we can study, for example, the glycosylations of ACE2 in mouse, in chicken, in pig, and find out why maybe the glycosylations of these species will give us guidance of how come some are more susceptible than others, because the sequence really is not showing that, it's not giving us any clue and why they're susceptible or not. So what I've tried to do today is give you a glimpse of why we think glycobiology of SARS-CoV2 and its receptor will be important for vaccine development and also for therapeutics. For the vaccine development, I think a lot of vaccine candidates are being processed right now, but I hope they take note of the fact that few glycosylation studies are now presented of the spike protein, and all of them look slightly different. They are all in HEK293 cells, but some showing mostly complex types, some showing some sites are glycosylated, some sites are not glycosylated. We are showing that individual glycosylations of the S1 subunit and the S2 subunit leads to more high mannose on the S1 subunit. That could be important if they're just producing the S1 subunit, manufacturing the S1 subunit, so it will be mostly high mannose types. How would that complicate, or how would that play around with the vaccine development, and what would that do, having the high mannose there? And the other thing that I want to point out is the O-glycosylation. I think when they've expressed the the trimer, they hardly saw any O-glycosylations, but we've seen these O-glycosylations when only the S1 subunit is expressed, and so there is an O-glycosylation there that could have an effect because it's closer to that binding region. On the S2 subunit, I think what I've tried to show that it is all sites are glycosylated, and this N90 site we think is important because it's where the RBD region is binding, and it's the only site that is showing some indication of susceptibility, differences in susceptibility between the different animals. So if you do have the N90 glycosylations, you're more susceptible. The other interesting thing is this N protein. I will not go into this N protein today, but we are currently studying this N protein. It's actually the most prevalent protein of the SARS-CoV2. It is more prevalent than the spike protein. And there was no previous study of glycosylations of the N protein. It was believed to be a phosphoprotein, but we've just recently started studying this N protein. It is more conserved, so it is less mutated. So it could be important for vaccine development because it has a potential, since it is more conserved, less mutated, it may be more important for vaccine development in the long run. We've shown that it's glycosylated, it's actually heavily O-glycosylated. So that could be interesting in the long run, looking at this as a potential vaccine, maybe in conjunction with the S protein. So that's something to look into in the N protein, but it's a complicated protein with number of O-glycosylation sites and also phosphorylation. So that will come in the next few months. These are the people who really helped to put this study together in the last few months. Everybody in the team really pulled together to be able to get this data out. Asif Shahjahan, Stephanie, Nitin Supakar, Anna, and Christian Heinz, they've all pulled together. I'd like to thank the NIH for the mass spec instrument, funding the mass spec instrument, and the Center for Plant and Microbial Complex Carbohydrate at the center has been a long-standing funding for us to be able to, for us to have the infrastructure support to do this quickly. because we don't actually have a direct funding to do this. And I'll be happy to answer any questions.